Okay, so the first category for lipids were fats, or they're also called triglycerides, because remember they look like the letter E with a glycerol molecule and 3 chains of fatty acids coming off of it. And remember that looking at that construction with the 3 fatty acids, that where the fatty acids attach to the glycerol, 1 water is lost, so you get 3 water molecules lost to make 1 fat or 1 triglyceride. And what makes 1 fat molecule differ from another fat molecule is the composition of these fatty acids. So remember, these chains are hydrocarbons, and meaning that they're made up of carbons and hydrogens. How can fatty acids differ? Their saturation, so they have double bonds or not. So if they have double bonds, they're considered unsaturated fatty acids. If they do not have double bonds, they're saturated. So whether or not they have double bonds can vary and make the fatty acids different. What else can make a fatty acid differ from another fatty acid? Yeah, the number of double bonds. So so they might have two fatty acids, both might have double bonds, but they might have different numbers of them. So Isabel talked about monounsaturated fat. Mono means 1, so that means 1 double bond. Poly means many, so polyunsaturated fat would have many double bonds. And so how many of those double bonds, even if they're both unsaturated, the fatty acids. And then also it could be due to the length, so you can have different lengths of carbon. So some fatty acids are longer than other fatty acids, and so on. And so that's another.
What's it called when there's 2? Is it di?
Yeah, usually you can call it that. Yes. Usually.
2, or is it just
yeah, you don't
poly?
hear. Yeah, usually you hear just poly unsaturated. All right, fun. All right, so let's look at phospholipids. This is category number 2 for lipids. This is very similar. Phospholipids are similar in construction to the triglyceride that we talked about yesterday. So, but instead of 3 fatty acids, notice that there's only 2, and the phosphate group, it says, is attached to a glycerol. So the phosphate group, so what does that mean? It means that instead of 3 fatty acids, I'm going to erase 1 fatty acid there, and in that spot to that carbon, and it usually comes up this way, is a phosphate group, and that's the PO4. That's one of the groups that we talked about with the functional group. And so so that's the different construction. So that's why I said they're pretty similar in that. Now the 2 fatty acid tails, these 2 right here, are hydrophobic because they're carbons and hydrogens. Those are nonpolar bonds. But because you add the phosphate group, and we'll see that sometimes the phosphate group can have other chemical groups attached to it, form what's called a hydrophilic head. So let me, if I look at, move this over a little bit so we can follow me. If we look at this part right here with the phosphate group, this is often called the head of the phospholipid, and the head is hydrophilic. And remember that it's because of this functional group here, the phosphate group. Remember, functional groups add polarity to the molecule, and so therefore that head is polar or hydrophilic. These fatty acids right here, they call these guys tails. These are the tails of the phospholipid. These guys are hydrophobic, and that's because of those carbon-hydrogen chains are nonpolar. And so that's the construction of that. So this picture here shows you a little bit more in depth about, instead of my generic little picture here, it shows you, here's the glycerol molecule. All right, so they're showing, you see the dot, 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 so they're showing you on each one of these models here. This actually shows you the atoms. So here's your 3-carbon compound with the oxygen. This is where the glycerol binds to the fatty acids, and so this is the double bonded oxygen of the fatty acid. So this is your carbon hydrogen. These are hydrocarbon chains. Notice here showing you a double bond there between the two carbons in that chain. And notice there, we talked about yesterday, wherever that double bond is, you get a kink in the chain, and so that's what they're showing you here. And those are your two tails, so these are the tails. And then off of that third, instead of having that third fatty acid, we have the phosphate group. This is what adds polarity, all right? And that's why we have a hydrophilic head there. This is the attachment. This part right here, this attachment here, this can change between phospholipids. So it's one way that phospholipids can differ from one another. That's just a chemical attachment there. So in general, all phospholipids have the glycerol, the two fatty acid chains, and a phosphate group, and then that can vary. All right, and so oftentimes in your textbook, they'll draw and illustrate phospholipids looking like this, okay? And as we get into, in a couple chapters, we're going to talk about the cell membrane. The cell membrane is made out of phospholipids, and so when we see a drawing like this, they're assuming that you remember why it's hydrophilic, because it has that polar functional group, and why the tails are hydrophobic, and that they're hydrocarbon chains. And so, so this is the cell membrane, and so this these are make up the cell membrane. Hydrophilic heads versus hydrophobic tails. Which one is water going to be attracted to? Which end of the phospholipid? The head or the tail will water be attracted to? Madeline?
The head.
The head. So water is going to be attracted to this part. Water is not going to be attracted to the hydrophobic tails. So they're going to want to, because of the charge, the hydrophilic head has a charge, and so does water, and so they'll be attracted. So what happens is, when you put phospholipids into water, I'll draw over here again. When you put phospholipids into water, so let's pretend like the board is a bunch of water here. What'll happen is those heads there'll be an attraction between the water and the heads, not the tails. And so what happens is phospholipids will actually form a little sphere in water where, and I'm not going to draw it all, but a little sphere in water where the phospholipids will orient themselves. So, because the heads are attracted to water, the tails are not attracted to water, and so the water is more attracted to the heads than the tails, and so the tails are oriented and get away from the water. So they actually form what's called a bilayer, bi meaning two. And so it has this bilayer, and this is the construction of a cell membrane. It has a phospholipid bilayer. I don't know if you remember that a little bit from bio. And so it has that phospholipid bilayer, and that's why it's constructed that way. And these phospholipids aren't bonded together in any way. They just orient themselves because of the polar nature of water being attracted to the heads and not the tails. And so that's our lipid bilayer and cell membranes. And phospholipids will behave this way, in the laboratory as well. If you put phospholipids in the water, they'll form little spheres, all right, because of that. So let's write, the next few bullets are about that. So when phospholipids are added to water, they self-assemble into that bilayer. So notice self-assemble, no one has to, it doesn't require any extra energy or anything like that, with the hydrophobic tails pointing towards the interior, like I drew here. And so this results in that bilayer found in cell membranes. And phospholipids are the main component of cell membranes. The majority of the cell membrane is phospholipids. We'll see later on in the course that there are other things that are embedded in the membrane, but phospholipids are our main type. And so this picture shows a little bit better than my picture there. It shows you more of the sphere. So it's not just a two-dimensional, but it's like a sphere that forms. All right, and that's phospholipids. That's category 2 of lipids. So the last category of lipids are called steroids. And steroids have a very unique structure, different than phospholipids and triglycerides were similar. This is very unique. So they have a carbon skeleton consisting of 4 fused rings. You'll see that in the picture in just a second. cholesterol is an example of a steroid, a very popular example. It is important because it actually also is found in animal cell membranes. So we have cholesterol in our membranes. We'll talk later on in the course about the role of that cholesterol in the membrane, but some cholesterol is good. Although cholesterol is also linked, if you have high levels in your blood, is also linked to cardiovascular disease, much like what other molecule was linked to cardiovascular disease? Saturated fat. All right, absolutely. All right, so this is the main construction of a steroid. This is the 4 fused rings. So here's ring 1, 2, 3, 4. Remember that they're not showing you every single atom in here to make it not as busy. So at each point is a carbon atom, and then the rings share sides, hence the fused part. All right, that's different than a disaccharide. Like when we talk about carbohydrates, they are linked like this. Notice that they're not sharing a side. So this is different. These guys are sharing a side. So that's what the fused part means. And so what they are showing you are these chemical groups, these functional groups, hydroxyl, methyl, all right, and so on. And then here you have a hydrocarbon chain. Each point here is a carbon. It's made out of carbons and hydrogens, and so that part would be nonpolar or hydrophobic. And that's what the main construction of a steroid is. This is cholesterol. But what you have different types of steroids, like testosterone and estrogen. They're also steroids that have 4 fused rings, but they have different jobs than cholesterol and estrogen and testosterone. All have different functions, and what makes them different, while they have all 4 fused rings, they have different locations of double bonds. So where the double bonds are, they have different groups, chemical groups coming off of it, so different functional groups and so on. They have different chains coming off of it, and that will give it different properties and therefore different functions in the bodies. But all steroids have this main construction of that 4 fused rings. So that's the only molecule we're going to talk about that has 4 fused rings. All right, so it's unique to steroids. And that's the last of your lipids. All right, I am going to do part of the proteins also today to give you some background information so that you can do group 1, start to look at group 1 in your pattern matching thing. So proteins are, we're back to that they are polymers. They're chains of smaller molecules. You guys remember what they're chains of? Amino acids. All right, so we're going to look at the construction of amino acid, what it looks like, and so on, and just some basic information about proteins. So notice here it says a wide range of functions. There are thousands of proteins, and they vary huge in what they can do. So there's quite a few different functions of them. So, and proteins are a big portion of the cells. It says accounts for more than 50% of the dry mass. Dry mass is if you take out the water, all right? And so if you look at other molecules besides water, proteins are a big portion of the cell. And so you can see that they have all these different types of functions. And so I just included this table here just to give you an idea of the types of functions. And I just want to briefly just talk about each one of them. So we've already talked about enzymes. So enzymes are proteins are all enzymes are proteins, and so enzymes we talked about in terms of speeding up chemical reactions. So there's enzymes in your digestive system, but enzymes are in every single cell of your body. There's thousands of enzymes that catalyze different reactions every second. Structural proteins, it says support. So some examples there. We have collagen and elastin in our connective tissue that helps to give us shape and support to our body. Keratin is a protein that's found in the horns of animals and so on to give its hard structure there. Silk fibers help with cocoons and so on. So those are examples. Storage proteins. Some proteins store, it says here, storage of amino acids. So amino acids are used to build proteins. And so the example that it gives is egg white. So like if you think about a chicken egg, the whites in there, the white part is basically all protein. And so what that does is it's a storage of amino acids for the growing chick, and they can use those amino acids to build proteins and grow. All right. The yolk part of it, the yellow part of the egg, is mainly fat. And they use that as well for energy and so on for the growing chick. But we're talking about proteins here. So egg whites, milk has proteins. There's proteins in seeds. Then transport proteins. An example that we'll talk about several times in this class is hemoglobin. So hemoglobin is a protein that's part of your red blood cells, and hemoglobin helps to transport oxygen. So when your red blood cells go up to your lungs, oxygen goes from your lungs and binds to the hemoglobin protein in your red blood cells, and then it travels throughout your body, and the oxygen goes to the rest of your cells in your body. So that's an example of a transport protein. Hormones. Some hormones are proteins. Hormones in general. Hormones are made in one part of your body, so an organ in one part of your body, go into your bloodstream, travel to another part of your body, and tell another part of your body what to do. So it's a way for cells to communicate long distance. So cells can't talk, but they can send out chemicals. And so one cell will send out a chemical, it'll go to another cell and something will happen in that cell that wouldn't have happened without that hormone. So those are hormone proteins. Insulin is an example of that. So insulin is produced by your pancreas, but is put into your bloodstream and travels to every single cell of your body through your bloodstream. Insulin is really, really important to be made by your pancreas, because without insulin, glucose can't get into your cells. What do we use glucose for? Energy, right? So therefore, if you don't have enough insulin, your glucose stays in your blood and never goes into your cells, all right? And that's bad news, all right? So that's an example of being produced in your pancreas but used all over your body. Receptor proteins, this kind of goes with hormones. So how does, if your insulin is made in your pancreas and goes to all cells of the body, how does it help to get glucose in there? And so sometimes, in your cell membrane, not only do you have a phospholipid bilayer, you also have proteins. And the hormone, like insulin, can bind to that protein. It changes the shape of the protein and then allows glucose to get into the cell. And so therefore, without insulin, the protein wouldn't allow glucose into the cell, and then that would be a problem. And so we'll talk later on in the course that is the issue with people who are diabetic, is that if they don't produce enough insulin, that's the problem, all right? Not getting glucose into there. Contractile and motor proteins, so actin and myosin are good examples. These are in our muscles, our skeletal muscles. They contract and help your muscles to contract for movement. So those are proteins, very important in that. And then lastly, defensive proteins in our immune system. You've probably heard of antibodies before. Antibodies help in your immune system attack against foreign objects, that are foreign particles. So that could be a bacteria, it could be a virus, it could be even pollen, all right? Anything that you breathe in or take into your body and get into your body that's not supposed to be there. All right, so that gives you just a brief overview of all the different things that proteins can do. So lots of different functions kind of all over the place there. I want to spend a minute and talk specifically about the enzyme one as an intro, because enzymes are very, very important, that we're going to build on this in a little bit in a few chapters. So they act as catalysts, enzymes, speeding up chemical reactions. A catalyst is anything that speeds up a chemical reaction. So a catalyst could be heat. Heat speeds up chemical reactions by you increase the kinetic energy, which means the molecules collide more, which means more reactions can happen. Enzymes are biological catalysts. They speed up reactions in living cells. So enzymes can perform their functions repeatedly. That means that they can be reused. So you have enzymes that can speed up one reaction and then go speed up another reaction. It's not used up in the reaction. And so this picture is giving an example or a picture of what an enzyme is doing here. So let's look at this enzyme. This is the big purple thing. All right. And so enzymes are chains of what, did we say? Amino acids. So when we look at this shape of this enzyme, why it has a shape, if it's just a chain of amino acids, like one amino acid hooked to another amino acid, etc. If we just look at just a straight line as a chain of amino acids, it really is not just linear, it actually folds on itself, and so you might have a chain of amino acids that folds like this, and so this is like the chain of amino acids, and that's what this purple blob is supposed to be showing you. Now in those foldings and so on of our chain of amino acids, you get a specific shape, and what's important about this picture is this part of the shape of the molecule. This part here, notice that the enzyme, it's called, and this is a typo, I'm going to have you fix it, it should be sucrase. Ace at the end of something means enzyme. So if you see ace at the end, it means enzyme. And notice that this enzyme, it's going to work, it's going to break down sucrose. Sucrose is a disaccharide. It's made from a glucose and a fructose hooked together. We cannot use sucrose, like if we eat sucrose, we can't use it in that form. We have to break it down into its monosaccharides. Hence the enzyme sucrase. So we have sucrase in our digestive system to help with that. Notice the shape of this enzyme and the shape of sucrose. They fit perfectly. So that's why this shape is very, very important. If you change the shape of this enzyme, it won't work anymore because the sucrose won't bind to it. And so the shape is important there. Notice here, from this picture to this picture, after they bind, the bond is broken, right? So here's our bond is broken. And this enzyme helped that to happen also with the help of water. What is water coming in doing here? What does water have to do with the breaking of that bond? You guys remember? Opposite of dehydration. Hydrolysis. So it's going to add water. It's going to split. That's the lysis part using with the help of water. So this is going to be used in hydrolysis. This is a little thing from the beginning of the chapter, meaning that it's going to add an OH and an H onto there. So therefore breaking the bond between the two. And then notice here now instead of sucrose, what's released from here is a glucose and a fructose. And they highlighted these guys here because that's what came from the water. All right, and so now you have glucose and fructose, and now we can use these guys for energy. All right, so used for energy. And notice that the enzyme can be reused. All right, so now can be reused to break down another sucrose and so on and so forth. So that's one example of the function of proteins. This is an intro, and we'll get into a little bit more detail later. All right, so proteins, as we said, are chains of amino acids, no matter what function a protein is, whether it's acting as an enzyme or something else. And so polypeptides, poly means many, peptide is referring to amino acids. So, so we usually call chains of amino acids polypeptides. A protein, how this relates to a protein, consists of one or more polypeptides. So the protein that we just looked at with the enzyme, I showed one amino acid chain all bent and folded to give it its three-dimensional shape. There are a lot of proteins that only have one polypeptide chain, one chain of amino acids. But some proteins have multiple chains of amino acids. Hemoglobin is an example. And hemoglobin actually has 4 chains of amino acids that all have to come together. And if you don't have those 4 chains of amino acids, you don't have a full normal hemoglobin molecule. So not all proteins are just one chain. So, but all proteins are made of amino acids, and so that's why the monomer is an amino acid. And I want to talk a little bit about the construction of the amino acid. This should be a little bit of a review because I already talked about this with the functional groups a little bit. So amino acids, remember the name, amino because it has an amino group, acid because it has a carboxyl group. So again, you should be getting to the point where you can see carboxyl group and go, oh yeah, that's a C with a double bond to an O and an OH. That gives it the acidic property. An amino group, oh yeah, that's NH2. All right, and that gives it what kind of property, the amino group? Yeah, it acts as a base, absolutely. And it acts as a base by, this can accept H pluses by acting it. Accepts H pluses, so it can take up H plus, which then reduces the hydrogen ion concentration, which is what a base does. All right, so amino acids differ in their properties due to differing side chains. These side chains are called R groups. R stands for radical. And so I'll illustrate this in these next set of pictures here. Whoops. And there are 20 total amino acids to form thousands of proteins. I use the analogy with words. Same idea with our alphabet. We have 26 letters in our alphabet, right? How many words can we form? I don't even know, thousands of words, right? And so the same idea with the 20 amino acids. You have 20 amino acids, but it can form thousands of proteins. With our words, it's the order of those letters that make words, the length of the words, it can vary and differ. Same thing with the amino acids. It's the order of the amino acids that determine the function or determine the protein and therefore the function of the protein. So this is an amino acid. So here's your carboxyl group and your amino group. When you're looking at for an amino acid, the carboxyl and the amino group, you are always across from each other. So you always have the central carbon atom. Here they call it the alpha carbon, and so you have an amino group and the carboxyl group attached to each other. If the carboxyl group was up here and not across from it, that would not be the construction of amino acids. You would not have that. All right, so it has to be across from each other. So you have 1, 2, and then the third thing is the third bond from the carbon has to be a hydrogen. This part right here is the same for all 20 amino acids. Right, this will look the same. What varies is this R group. And this R, I'm going to put here group here. This R is not an atom. There's not an element R. All right, so the R stands for the R group. And this is the part that changes from one amino acid to another. It makes the 20. So all 20 different amino acids, there's 20 different R groups. And so that's what these set of pictures here, they have a couple of different charts here that just illustrates this. You do not have to memorize these amino acids, all right? You just need to understand and be able to identify what you're looking for an amino acid. So let's look at this first one. Well, actually, let's look at the first one here. Amino acid, we just said, has an amino group and a carboxyl group across from each other. I want to point out here how the textbook wrote those. This right here on the left is the amino group. Notice it has NH3 with a positive. We just talked about that, so here's N. So if we were to draw out here, amino group is NH2 attached to, so this is what it should look like here. If this says NH3 with a positive, that's still an amino group. Can somebody tell me why?
Since it acts as a base, it can pick up an H plus ion.
That's right. So this is still an amino group. Let me get a different color here. But it has already acted as a base. Amino groups act as bases. Excuse me. And when it picks up that H plus, it makes it NH3 plus. All right. So I want you to recognize that this is still an amino group that's already acted as its function as a base. And so then the carboxyl group on the flip side here. The carboxyl group, we've learned, is C with a double bond to an O and an OH off of that same carbon. And so this is O negative. What does the carboxyl group do? It acts as an acid, and acids do what? They release H plus ion. So what happens is this has acted as an acid, releasing a H plus ion. The H plus is positive because it left its electron behind, so therefore that's where that negative charge comes from. So this is still the carboxyl group that has given off already its H pluses and act as an acid. So I wanted you to be familiar that those are two different ways to draw those functional groups. All right, so if you cut off the tops here that are highlighted in white, every amino acid looks the same, has that amino, hydrogen, and carboxyl group. So what's highlighted on the top are your R groups, and your R groups can change. So you can see the H, CH3, and so on and so forth. These guys are classified as nonpolar amino acids. That's because when we say nonpolar, what you're looking at is your R groups. Your R groups are nonpolar. So if you look at the R groups, they're hydrocarbons. They're made out of just carbons and hydrogens, roughly, and so it makes them nonpolar, so those amino acids are classified as nonpolar. So when you're looking at that they're looking at the R groups. Now, every amino acid is somewhat polar because it has the amino group and the carboxyl group and so on and so forth, but the classification is based upon the R group. So if you look at polar, again, looking at the R groups, they have functional groups. So you have hydroxyl, sulfhydryl, amino, and so on, and that adds polarity. Those are polar, and so adds, it makes them more water soluble. So those are your polar R groups. And then you have electrically charged R groups. Notice here that these R groups have either the carboxyl group or the amino group here. And so they have charges here. So these guys have the carboxyl group that acted as an acid, so they have a negative charge, the R groups. These guys have the amino group that acted as a base and are positively charged. That's why they call it electrically charged. And one other thing about amino acids is usually you'll see them shorthanded by the first 3 letters of their name. And so instead of writing the whole thing out, you'll see the first 3 letters. Okay? And you don't have to memorize the names and all that kind of stuff. You don't need to know all that kind of things. All right. So then we hook amino acids together to make proteins. They are linked by these bonds called peptide bonds. And a polypeptide is a polymer of these amino acids, and we hook them together. And they can range in length from a few to more than 1,000, so it depends on the molecule. And each polypeptide, what makes them different from other polypeptides is that sequence of amino acids. So is it glycine next to alanine, next to arginine, and so on and so forth. So how they're hooked together changes the construction. And so we study proteins and figured out, like we know the sequence of amino acids with all 4 polypeptide chains in hemoglobin. And so how we do that is in the lab, and now most steps in sequencing and figuring out the order of amino acids and proteins so we can study them is automated. We have chemical programs, computer programs that will allow us to do that. And so then once we know the order of the amino acids, then we can look at the structure. So a functional protein like hemoglobin consists of 1 or more polypeptides. So hemoglobin has 4 polypeptides, and they're twisted and folded and coiled into a unique shape, like I did with that picture of the enzyme. So they have a particular shape that allows it to do a particular function. And why it has that shape is the sequence of amino acids. And that sequence of amino acids determines its 3-dimensional conformation. Your book uses this word a lot. Conformation means shape. So it has a 3-dimensional shape. So what happens is that sometimes if you change the order of the amino acids, it won't twist and coil the same way, which will change the shape, which will make the protein not work. All right. So the order of amino acids is really important so that they twist and coil in the right way, so they do have the right shape. So like for instance, the enzyme sucrase has the right shape to bond with sucrose. So it's a protein's conformation or shape determines its function. So it's very, very important. And what I tried to draw with the enzyme was like a ribbon model, but there are different ways that biologists try to show a protein shape, and these are two examples here. So here's the ribbon model where the ribbon is like that chain of amino acids that I kind of drew with the enzyme example. Sometimes they'll just show how the atoms are bonded in what's called a space filling model. This kind of gives you the overall shape of the molecule, and so for instance, like this shape here, this right here might be part of its active site. All right, where that's where the sucrose binds to the enzyme, and so that's another way to look at it. So you can see them drawn in two different ways. They're both showing the same thing. Here's that same groove. All right, so if we just do the overall shape here, the overall shape would be the same. They're just showing the atoms and the connections differently. All right, and this is where I'm going to stop, because I'm going to give you the rest of the time to work on that protein matching. All right, or not the protein, but the pattern matching. That protein's on my mind. So 3 and 4 are due tomorrow. So I would start there. All right, and so if you have any questions on that, and then you can go back to 1, which is about proteins. 2 is about steroids that we talked about today. The only one we haven't talked about is group 5, which is the nucleic acids, which we can talk about tomorrow. All right, so I do have up here, if you get to the cutting out part of it, I have some scissors and tape. In my tape thing, I have masking tape. Don't use masking tape. I have enough, like, Scotch tape. I have tears. All right, and so if you have any questions or we can work together. Oh, yeah. Sorry.
I answered the question,
and you guys are looking at all the pages, like that second packet with all the pictures of the molecules. You're looking and flipping through all of those.
In this example, all the carbons are attached to an oxygen, but in this one is like
So the location of that's somebody else does that.
and then
it's asking how many bananas do you have to
I was here because a single string monosaccharides.
Because remember, like monosaccharides. So remember okay. Find any of the monosaccharides like
String like that.
I didn't except that there's no monosaccharides they don't have
Aren't they like they are? I don't think cheese just
So the things for us to look for,
So you should have, one,
8 out of just right? Two rates put together.
I thought straight cheese was put together like this. Why is that? Yeah. Oh, yeah. I like that. Like, yes, I can see it. And then 2 single that. And then it's
amino acids, not fatty acids.
They don't all have
It's
to
okay. It's okay.
So remember, let's figure it out. Yes. So remember,
amino acids are
so therefore,
it's like carbon and like,
yeah, like if they're not sharing a side.
I see.
All right, and so they're bonded, and bonded.
Okay, so because like
So that would be a disaccharide. So monosaccharide would just be one of those. Right? So this would not be a so this would be a monosaccharide. This is a monosaccharide, even though it's the same, because we can see here that it's nitrogen. So it's carbohydrate. Right, but when they go from the air to that double bond,
A lot of them are like a seed that works on
so they're single bond.
so this is it.
Yes, there are.
Do you mind if I like copy this?
because it adds
Thank
No, it's all in the Yep, you got it.